Welcome to our new format of panel discussions presented by Eppendorf with our today's panel topic, Rethinking Vaccine Development. I am Stefan Goldenbaum, your moderator, and I will guide you through the panel discussion for today. I welcome our three guests, Jörg Schwinde, Segment Manager at Eppendorf and expert in vaccine development, as well as monoclonal antibodies. Also live with us, Tim Schwamz, Global Automation Marketing Manager at Eppendorf and responsible for vaccine development from an aspect of automation, as well as our third guest. Please welcome Dr. Julian Lopez from ContiVir, a project from the Max Planck Institute for Dynamics and Complex Technologies. Welcome, all of you. Vaccine development is a topic with multiple facets in terms of production, research, development from different kind of angles. That's why I would like to start here with Jörg, first of all, giving us a little bit introduction about himself.
Yeah, all right. Well, I'm responsible for the segments of vaccines and monoclonal antibodies, as you have explained. My background is microbiology and molecular biology, and this function was created quite new. It exists for 1.5 years now.
Thank you. You are specifically dealing with the topic of vaccine development. Which kind of vaccine development methodologies do you see?
Well, fortunately, there are several ones available. This does mean that there is not the one golden way to do it. And let's start with the assumption that a pathogen, or at least a part of a pathogen, a unit of it, a protein component, a sugar component, or something like that, is available. You can start with the development, and this is possible to do with the whole complete pathogen, which can be a virus, which can be a bacterium, which can be a protozoan, with protein subunits of it, or even down broken to the nucleic acid area, with which you can work and start to develop vaccines.
When you look at this very, very rich field in vaccine development, do you see here any geographical hotspots in terms of the research, or do you see a centralization of the research into certain areas?
Yeah, centralization would be a little bit over-mentioned. Though we can observe that there are very strong actors in the global vaccine research and development and manufacturing in Europe. So we do see that about 70% of these actors spend their R&D budgets here in Europe. 80% of their capacity is manufactured in Europe, and 86% of these are exported globally. Though we need to see, and thankfully, that there are strong activities in the Americas as well as in the Asian countries. Looking at the real global export factor, there is a focus on behalf of the Asian countries on defined parts of the world. Not urgently, their products are exported worldwide.
When you say there is not a total centralization on Europe, but nevertheless also you can say a significant kind of exposure of Europe here, you don't really see a cost factor coming into the whole game.
The cost factors do play a major role, because we are also talking about the accessibility, availability to treatments and therapeutics worldwide. And certainly there is a difference between developing and developed countries. This will be supported by local manufacturing, certainly, of course. But thankfully, we can also see that treatments are supported. So even in countries where the accessibility by finances to treatments should not be as advantageous as in other countries, there are organizations like the WHO or foundations, famous foundations, we know the names of them, who do drastically support this. So the manufacturing factor and the costs for this is the one story. And this might be one of the reasons why from some countries or on behalf of some countries where we know about big and huge manufacturing facilities, that they are not specialized on international, global exports, though, of course, on international exports as well, in defined territories of the world.
When we leave from a research and development point of view, the regionalization and also the cost factors a little bit aside here, and speak more about the players in the market, do you see, when we talk only about the main players in the area of vaccine development, see here a concentration to a few global, or is this also a field which is covered by a huge variety of players in the market?
Yeah, there is a variety in the vaccine market, certainly, and this is very good. By revenues, there can be identified 4 strong players with European and American origin. But as I say, this is by revenue only. If we look at doses, the most successful one is coming from India.
Mm-hmm. When we speak now about development, and you also mentioned you more or less see 4 main driver here in the market very present, is there anything that is very, very important for them in terms of development, production, which really drives them to become such big players?
Yes, sure. Well, it is the product itself, of course. It is the mode of manufacturing. It is also the help. We have talked about the cost factor, which is always there. And one of the important and relevant supports in this concern are the facts of automation.
Mm-hmm. Thank you for your answers here. When we talk about automation, that brings us directly to our next panelist here in this group, Tim Schwamz. I introduced you briefly in the beginning. Maybe you can say a couple of words about yourself and your work and how this connects to automation and vaccine development.
Yeah, thank you for this kind introduction, and thank you again for inviting me for this panel discussion and letting me join from my home office here. As already pointed out, I am responsible for the automation portfolio here within Eppendorf, which covers a variety of devices from smaller scale to larger scale, which can be used for also a variety of different workflows. And among those workflows, very important, and also the reason why I'm here, is the vaccine development workflow. And from my background, so I have a background in molecular biology and virology.
Thank you, Tim. And when we directly dive into this topic, what do you see, or how do you see the role of automation in vaccine development?
Yeah. So let me pick up on something that Jörg said, and obviously automation is very important at the, let's say, last steps of vaccine development and production. And here we are talking about huge assembly lines, huge automation solutions for the formulation of doses, for filling doses, and also for packaging. So very much in the field of production scale. But there is a certain trend as well those days, and this is that automation is also getting more and more important at the very early stages of vaccine development. So at the screening stage, at the actual finding of potential vaccine candidates, at the testing stage of those candidates on molecular or cell biology level. So what one can say is that automation is now, or can be, part of the entire vaccine development workflow, and different devices and different sized automation offers different options and different degrees of automation for the entire workflow.
Do you see in this connection here also pros and cons when we talk about automation?
Yeah, that's a good question, and it's actually a question that if you work in automation, you get quite frequently. So the obvious pros for automation are number one, productivity / throughput. I mean, that is something that is very obvious. If you get to an automated solution, it mainly means that you can achieve a higher throughput, that you can process more samples, more screening candidates, that you can go through much larger screening procedures within vaccine development, for example, as you can simply handle also 384 well plates and so on. But I personally think that there is another aspect of automation which might be the biggest pro, and that is the term of reproducibility. That means that if you have a process within an automated setting, this process will always be performed with the exact same speed, with the exact same parameters. So the results you get at the end of this process will be extremely reproducible, no matter which day you do it, no matter at which time you do it, and no matter who is actually operating the automation device in this respect. So and in addition, with this reproducibility, that there are coming some other things connected to it. So if we think about documentation, of course, this is easy if this has been done in an automated fashion, and also time saving, not only due to the productivity aspect, but also due to the fact that important workforce has more time to do different things because the automats are taking over certain steps of a process. Cons, I think also very obvious, is that automation is usually a big upfront invest, not only on a monetary point of view, but also if we talk about time and capacity, because it takes some time to get a system up and running. It also takes some time to get everyone trained on that system. This is going to be compensated over time, but in the beginning, it is a hurdle that needs to be overcome. And to be precise, one has to say that not every process is equally suited to be automated.
Yeah. When we talk about this quickly, and you mentioned the difficulties, and you also mentioned a couple of cons here. Are there any differences from your experience in the level of how certain processes are automatable?
Yeah, good that you're picking up on that. Very good point. So let me start with the bits that are easy to automate. So generally speaking, it's usually best if you start automating a a process which is already established within your working environment. So if there is a SOP already existing, or if there is a good protocol already in place for a process, so those are the processes which are much easier to automate or to transfer to an automated process as compared to something that maybe you've never touched before, right? So if you start with something from scratch and you want to get started with the process and automation, it's not impossible, but it is usually more challenging. So automation favors standardized step-by-step processes, which can have varying parameters. So that is not a big deal. But those are things which are suited for automation, and that brings me back to something that Jörg said earlier, is that the current development that we see a push towards nucleic acid based vaccine candidates, I have to say, obviously that opens up a big space for automation, because if you look at the molecular biology bits of the vaccine development and testing, one has to say those are usually the ones which are much easier to automate, because there is a set toolkit, there is a lot of methods you can fall back to, there are a lot of standardized processes, there are not too many moving bits and pieces, so molecular biology is easy to automate. On the other hand, and that brings me to the processes which are not so easy to automate, everything that needs a lot of adjustment and handling and experience on the run are processes that are not so easy to automate. So here I'm talking cell biology bits and pieces, for example. Let's say something very simple, trypsinizing cells. So if you wash the cells and then add the trypsin to detach them for cell culture, and then what you usually do is you look at this cell culture flasks and plates, and then you give it a minute more or less, depending on the state of the cells. So that is a process which is not so easy to automate, because it needs someone to observe it and to adjust it on the fly. I don't say that it's impossible to automate, but usually it either requires much larger automation or much more complex automation solutions, or it involves more so-called interruption within the automation process, so more manual handling steps. And at some point, if you involve too many manual handling steps, it's questionable if that makes sense for automation at all. So as I said, like rough rule of thumb, molecular biology bits easy to automate. The cell biology parts within the vaccine development not so easy to automate, but possible.
You spoke a lot about scalability here, which certainly addresses a certain large-scale market when it comes to production and also to the research and development part. Do you see here also a certain trend towards automizing the mid and small level vaccine development? Or is that only something what you just described here for large level productions?
It's absolutely not only suited for, let's say, the top 5 vaccine developers out there. So during the last years now, we see a huge trend towards smaller automation solution, also step automation solutions, which have benchtop scale, so which fit most of the regular lab environments without big adjustments. So you don't need the extra room, you don't need some sort of like special table or stuff. Also, you don't, for most of those solutions, you don't need extra infrastructure, let's say high voltage, for example. So it's easy to to set it up, it's easy to install, and it's easy to get started with those types of automation. Of course, that also includes the software part, which is a huge bit of automation, right? So a lot of people are afraid that they have to learn programming, or that they only need that they need special stuff to operate the automation. So with most of the benchtop automation solutions, this is not the case anymore. Software is usually easy to handle. A lot of people can learn that very quickly. So a lot of people can work with this device. So that makes it obviously, combined with the comparably lower costs, very attractive for smaller scale labs, but also for spin-offs and startups which are focusing on vaccine development.
Mm-hmm. Thank you, Tim. When you just mentioned spin-offs and startups, then we have also to speak a little bit about new methodologies, new techniques, and that brings me to Julian Lopez from ContiVir. Also here, could you describe in the beginning a little bit for our audience, what are you doing, what is your company involved?
Well, thank you, Stefan, for the introduction and for inviting us, the ContiVir team, here to this panel. Well, ContiVir is a direct result of the doctoral research of two members of the bioprocess engineering group at the Max Planck Institute in Magdeburg. Mm-hmm. Currently, we're being funded by an Exist Transfer of Research program. We basically want to enable a revolution on the manufacturing of vaccines, of virus, for gene therapy and vaccines by providing the most efficient production and purification platform. And we're doing this out of previous infrastructure that we have already there. And well, I'm in charge of the business development of this project. I just recently joined the team.
Which development trends do you see from what you are doing at ContiVir in the vaccine research area?
So, for example, for some types of vaccines, we've seen a transition, an ongoing transition between egg-based towards cell culture-based. Mm-hmm. In some other vaccines, especially the ones that are already established as cell culture-based, we see that they are, the field is just implementing the same process intensification strategies previously used for mAb production, such as optimization of the expression cell lines, the modes of production, improving downstream, moving to perfusion, improving the cell culture media, and whatnot. Recently, we've also seen a trend towards at least more interest in continuous processes. These, of course, has all been because we want to tackle this challenge seen in manufacturing. And this is for the current situation, but from our perspective, what happens in 10 years, for example, when gene therapies move towards common diseases, not just the rare ones, and also with increasing population, what happens we're going to need more and more vaccines. And from our perspective, process intensification then must not only focus on the proven strategies as the expression lines or the manufacturing process, but on the process tools themselves, the technology, the devices. Mm-hmm. And in this part is where we see the important contribution, at least from our side, from startups, because they can take on riskier or more disruptive projects than the larger companies.
Yeah. Is that in general something which you would see more and focus, what Jörg mentioned previously, of the 4 or 5 big players, or is that something where startups become a game changer?
Of course, large companies are working and providing solutions for all these, let's call them challenges. But in some smaller projects, or at least the more disruptive ones, it might be more appropriate for startups to tackle these kinds of challenges. As startups, we can help reduce the uncertainty for everybody in the field. And the role performed by us might be different. I mean, larger companies play a different role in these developments.
And how do your solution affect these developments?
So, for example, our system, our production system, is based on a fully continuous tubular bioreactor for virus propagation and a size-based capture chromatography step for virus purification. Because another issue in the area is that we try to use technologies developed for vaccine purification or for some other type of molecule purification or production that are not entirely compatible with vaccines. And for example, these two technologies combined can attain a 10-fold increase in efficiency compared to current technologies, such as batch production or packed bed chromatography. So, well, other advantages, it's a fully scalable system, it's a single-use system, and we offer the unique advantage through our technology of reducing both the time and the space needed to produce large volumes of highly purified product.
Okay. When you think about the idea that comes certainly first to develop a certain technology until what you just described to have really at the end of the day purified product ready for the market, what is your estimation or idea on the time to market here, the period? How would you judge that?
By maintaining the efficiency across all scales, this is related to the scalable ability of our system, and minimizing and shortening the development time, we can minimize production delays for entering clinical phases, for example. Just as an example, we were working on an experiment with influenza with one of our prototypes, and we were able to reduce a 60-day current process, to just 27 days. Now imagine that being translated into a small company or a large company that a lot of people are working right now in gene therapy and in vaccines. Imagine having that advantage that you can match the output or even more in less than half the time.
Yeah. My last question, you have certainly a very good overview about, let's say, the research market here, also research projects in terms of diversification on certain, let's say, viral or even bacterial parameters, right? Do you see at this moment, due to the very strong focus on COVID vaccine research, any impact or any future impact on the future research and development of other vaccine generations for already existing infectious diseases?
We see it as a positive influence, let's call it, because it's highlighting the status of the vaccine field right now, all this attention because of the COVID vaccine. It highlights the status of the field. It also it shows the need of bringing flexible technologies to the field in order to be able to overcome this challenge. I mean, right now we don't need double or twice the capacity. We need 10 times the capacity. So from our perspective, and what we've seen the reports, I mean, more and more companies are bringing their know-how, their resources to try and tackle the challenge of the COVID vaccine. But we see that it's not going to stop there. It's going to be beyond that. So all of this fresh perspective, let's call it, into the field will start questioning, okay, is this the best way to go about developing this other type of vaccines? So yeah, it's a positive.
Yeah. Thank you, Dr. Lopez.
Thank you.
We have learned a lot about the current situation in vaccine research and development. We got a nice overview from a global perspective, from a provider perspective. We have learned about the impact of automation, and we got insights from a specialist from a small startup here about new trends and new possible developments in the future. Visit our website to learn more about bioprocess for vaccine development and other applications. I would like to thank all of you for watching. I also would like to thank our panelists for participating and sharing their knowledge and input here. I would be happy seeing you in the future for other panels we will provide, and thank you.